Okay, so good evening, everyone, and welcome back to today's Asset Colloquium, the online Asset Colloquium. For those of you who are joining from outside TIFR, ASSET stands for Advances in Science, Engineering, and Technology, and one of the main tasks of ASSET is to organize very interesting colloquia on the topics that I just now mentioned the fields about. And these are usually organized at 4 o'clock on every working Friday. Today we have yet another very interesting colloquium titled as Drawing is Inquiry by Dr. Ipsa Jain. And in fact, who kind of says images have been part and parcel in the progress of science, which many of us completely agree with this sentence. And also she is going to more interestingly show how scientists, artist collaboration can contribute to the scientific pursuit. And even more interestingly, she is going to kind of show this by using very interactive way of disposition. And so let us look forward to her very interesting lecture today. And before we go further, I would like to thank Arnab for making this talk possible. He actually proposed this particular speaker and also the topic and so on. And so therefore, I request Arnab to formally introduce today's speaker and then on to Dr. Jain.
Thanks, Satya, and welcome everyone to the Asset Colloquium. And thank you so much, Ipsa, for joining us online. For the topic that is there today, this is really going to be not as good as having her here and drawing and looking at things that we doodle with her is very, very interactive and fun. We are going to try and give you a little glimpse of that. So the first thing I'm going to ask everybody before I even introduce Ipsa is please go and get yourself a pencil or a pen and a piece of paper because this is going to be an interactive session. We are going to be doing some small 30-second doodling exercises is what Ipsa tells me, and you should be able to take a picture of it, and there is an email address. It will come on the slide. I'll put it in the chat as well. You take a picture and send it to us by email. So that's the plan of how we're going to try and make this a little bit more interactive than just a speaker on a screen somewhere. But it's a pleasure to introduce Ipsa. She is a scientist who has turned into an artist and an illustrator. And she started off doing biology, I think it was oncology at IISc, and then in a very remarkable postdoc period at INSTEM, she decided to look at scientific visualization in a lot more detail, and in so much more detail, and that she decided that this is what her career is going to be. And she moved over to the field of scientific illustrations and art, and looking at how art can be used to actually take science to the public. And which is how I know her, through these outreach efforts, that she's also had some fantastic efforts in illustrating things which are otherwise just so complex to understand. A figure is not just worth the 10,000 words, but much more, especially if Ipsa draws them. So I will shut up here and just say that if there's one word you should know, it's Ipsawonders. Okay? Ipsawonders is where you track her down. If you just Google for Ipsawonders, you will find all her work and all about her. And with that, it's over to you. And I hope, again, reminder, get ready to do some interesting exercises during the session.
Thanks, Arnav, and thanks, Dr. Satyanarayan, and good evening, everyone. I'm kind of sad that I'm not there with you today and we're doing this online, but maybe in future we'll get an opportunity to do this together in person. So introducing myself briefly as, just a second, let me get, okay, slide change is not happening. Just give me a second. Yeah. So I'm somebody who trained to be a scientist for a major part of her life and is still someone to the core. And except the tools that I use now are more like pencils and brushes instead of a pipette. And as a proof of my credibility, I'm going to give you a glimpse to my work. And allow me to also add a disclaimer that the examples I will speak about today will mostly be from the tangible world of biology. This talk will be part image display, part activity at your end, part reading, and a part scientific talk. And please, again, grab some pen and paper so that when the activity comes in between, you are ready to give it a go. Let's delve in. We are curious. We want to try things out. We want to get distracted and generally look like we are wasting time. And maybe we are wasting time right now. Sometimes we just want to see, to observe what is happening for the hell of it. To observe is to look at, listen to, touch, taste, or smell something, attending to its details, and take in the resulting perceptual experience. We gain useful evidence simply by noticing what's going on around us. There's a high concentration of nerves and finely tuned muscles in the palms and the fingers. As a result, we can perform very intricate tasks with our hands. The process of material and knowledge production is a single hybrid affair in which the work of the head and of the hand form a complex whole. From 16th to 19th century, the illustrated scientific book remained the gold standard of scientific visualization, like the detailed anatomical drawings in Vesalius' book, or the Euclidean drawings in supporting Copernicus' argument for heliocentric universe. The intricate drawings of the microscopic world in Hooke's Micrographia, geometric diagrams relating to fundamental laws of motion in Newton's Principia, schematics mapping the refraction of light rays through a prism in Newton's optics, drawings of scientific instruments, and tables systematically arranging the known universe of chemical elements and compounds in Lavoisier's elementary treatise on chemistry. Drawings of geographical strata that reveal Earth's history in Lyell's Principles of Geology, and floating microbes produced by fermentation in Pasteur's Studies on Beer. The 19th century also added a new modality to scientific books and journal articles: photographs, sometimes taken with the aid of a telescope or microscope. 20th century saw invention of image-producing research equipment, X-ray-based material analyzers, cloud chambers, particle accelerators, electron microscopes, satellite space telescopes, DNA analyzers, and so forth. In the late 20th century, the power of computer for visual representation of large masses of data was harnessed. This unleashed a wave of creative image making unprecedented in the history of scientific visualization. Image creation and analysis has always been at the heart of scientific discovery and dissemination. With built-in instrumentation, is the role of hand lost? Let's uncover. So this is our first activity. Please grab your pen and paper. What I'm going to do is read out a passage from this book that's there on the screen. And as I read out, please try to imagine what I'm describing and try to draw it as quickly as possible. I will read the passage twice for your convenience. Once you are done, please take a quick picture with your smartphones if you have access to one, and send it to this email ID, which is sciartspeaks@gmail.com. My friend Kadambari is behind the screens curating these images, and she'll put them together so that we can look at it at a later part of the talk. I'm going to begin. In an ordinary plain section of a vegetable parenchyma, we recognize the appearance of a froth, precisely resembling that which we can construct by imprisoning a mass of soap bubbles in a narrow vessel with flat sides of glass. In both cases, we see the cell walls everywhere meeting by threes at an angle of 120 degrees, irrespective of the size of the individual cells, whose relative size, on the other hand, determines the curvature of the partition walls. On the surface of a honeycomb, we have precisely the same conjunction between cell and cell of three boundary walls meeting at 120 degrees. In embryology, when we examine a segmenting egg of 4 segments, we find in like manner that the same principle is still exemplified. The 4 segments do not meet in a common center, but each cell is in contact with 2 others, and the 3 common boundary walls meet at the normal angle of 120 degrees. A so-called polar furrow, the visible edge of a vertical partition wall, joins or separates the 2 triple contacts and so gives rise to a diamond-shaped figure. I will repeat it again. In any ordinary plane, the section of a vegetable parenchyma, we recognize the appearance of a froth, precisely resembling that which we can construct by imprisoning a mass of soap bubbles in a narrow vessel with flat sides of glass. In both cases, we see cell walls everywhere meeting by threes at an angle of 120 degrees, irrespective of the size of individual cells, whose relative size, on the other hand, determines the curvature of the partition walls. On the surface of a honeycomb, we have precisely the same conjunction between cell and cell of 3 boundary walls meeting at 120 degrees. In embryology, when we examine a segmenting egg of 4 segments, we find in like manner that the same principle is exemplified. The 4 segments do not meet in a common center, but each cell is in contact with 2 others. And only 3 common boundary walls meet at the normal angle of 120 degrees. A so-called polar furrow, the visible edge of a vertical partition wall, joins or separates the 2 triple contacts, giving rise to a diamond-shaped figure. Now I'll give you a break of 1 minute to quickly capture your image and send it to the email ID, please. Thank you. All right, moving on. This is how some other people on a different day in a different room responded to the same passage. Each one of us is imagining objects and process in a different way with the clues given and projecting them differently. This is based on our experience with the tool and sometimes our prior knowledge of the subject matter. In his notebooks, he begins with the phrase, I think, and ends not in a sentence, but in an illustration. A tree that represents key concepts, descent, diversity, and extinction, the great tree of life. Visual displays are distinctively involved in scientific communication and in the very construction of scientific facts. Observations are fundamental base of all research methods. The beautiful images in Maria Sibylla Merion's Metamorphosis Insectorium Surinamesium are the product of meticulous observations of life cycles of insects. Most of her images were made from live or freshly preserved specimens. Marion shows moths laying eggs, caterpillars feeding on leaves. In order to create complex compositions containing a variety of organisms accurately depicted, she relied on individual studies made on small pieces of vellum. When an animal or plant was copied onto a larger composition, she changed very little, preserving the posture, color, and details of the specimen and its relationship with other specimens in the drawing. Seeing is an some respect, and art, which must be learned. In his preface to Micrographia, Robert Hooke wrote that all that was required for studying nature was a sincere hand, a faithful eye, to examine and to record things themselves as they appear. In this passage, he implies that with his words and his pictures, he acted as a microscope's assistant by simply recording whatever appeared through the lens of the instrument. Hooke's illustration of the ant, or the one on screen here, at a scale that is many times its actual size, highlights the insect's brittle structures and spiky limbs by silhouetting them against a stark white background, thereby effectively transforming the seemingly familiar ant to an exotic and mysterious alien creature. Hooke built up these images from numerous observations made from multiple viewpoints, multiple lenses, and under various lighting conditions. Vision is not a mechanical recording of elements, but rather the apprehension of significant structural patterns. In this micrograph, darkly colored cells in close-knit clusters punctuate an otherwise relatively uniform tissue. To make these images, embryologist Saunders immersed living early stage chick embryos in dye, Nile blue sulfate, which is a vital dye. It colored cells without disrupting their normal processes. After washing, the pigment remained only in particular groups of cells. He believed initially that these cells were precursors to melanocytes. Things changed when he mapped these blue cells in developing embryo. After staining and fixing tissue slices, he would trace the outer boundaries of the tissue marked in these images by solid lines, and the position of the blue cells onto the glass plate by dots. By lining these glass plates together in a sequence, he could see where the clusters of blue cells appear at one developmental stage. He figured these cells were not pigment cells, but in fact in the process of dying to part and articulate the digits and the limbs. We need to show the real world, not lines and numbers, as we strive to create visual solutions for visual problems. The image here is the painting of myoglobin, the first protein structure solved by X-ray crystallography. The painting was created by Irving Gies for a Scientific American article, The Three-Dimensional Structure of a Protein Molecule by John Kendrew. He created this painting by first photographing the physical models and then creating voluminous sketches and studies before painting this finished version. He made small adjustments to avoid structural overlapping. He might distort the protein a little bit here and there, or he might use a slightly different viewing angle or perspective for a different part of the structure. He wanted to draw the protein not exactly as it was, but to show how it worked. He called this process selective lying and claimed that this was one of the special talents of a knowledgeable artist. A computer could draw a protein given the right set of coordinates, but who would tell it exactly what to show? In 1968, Waddington tells us that science is something more than a collection of conceptual or practical results. It's also an activity, and its practice involves, as a very important part, the exercise of the faculties of insightful perception of natural phenomena and of the imaginative creation of new concepts. Image on the left is a top view of Wellington's epigenetic landscape, where the path followed by the ball represents a developmental trajectory. The valleys represent different alternative paths to depict different developmental potentials. For example, differentiation leading to different cell types. On right, by depicting the underside of the surface, Wellington illustrated the idea that genes can change landscape during evolution. The pegs at the bottom represent genes acting on the landscape by tugging on the intertwined web of ropes, thereby changing the topography of the valleys and ridges above. These images do something very well. They stimulate visual thought. To paraphrase Waddington himself, his artful representations help to loosen the joints of scientists' imagination creating a space for associative play required to introduce new concepts in theory formation. The world in Waddington's landscape remains same as it rolls down the valley. It does not undergo any morphological transformations. Artist Gemma Anderson and collaborating scientists imagined overcoming this limitation by replacing the ball moving through landscape with an isomorphogenesis transformation. Isomorphogenesis is a drawing-based series which explores the potential of representing morphologies as a dynamic and formative process. The resulting experimental image on the left reduces the level of abstraction of Waddington's representation by explicitly showing the trajectory of morphological change through the valley of the landscape. Creativity. The formation of new association and concepts requires playful abandoning of preconceived notions. To be creative, we must let our minds wander. They say hand drawing must not be allowed to disappear. They say this not because of its use in scientific illustration, but because of its potential to form sight, insight, and understanding, and to share this understanding, and to enhance creativity among artists and scientists alike. So let's try and be playful for a bit. So I'm going to give you certain word prompts. The first one is worm, and you have precisely 1 minute to draw the first thing that comes to your mind when you look at or think about or hear this word, worm. All right. I'll take you to the next word, glowing worm. You have 1 minute again. And once you are done, again, please capture your images and send them to the email ID sayartspeaks@gmail.com. Somebody, were you able to hear me? Somebody just said that they couldn't hear my audio.
No, we are okay.
Okay.
There's a problem. I would say, Alak, can you please check your audio?
All right. Please send your images to Sayat Speaks, where Kadambri is waiting to compile these images. Okay. And I'm just going to try and figure out how to show you what's already been shared. All right. So this is how we looked at the cells previously. Can you see? Can you see? Just if someone says yes, then we could. Interesting. Just give me a moment. I lost my presentation. Yep. Renaud, my collaborator said, drawing enables the creation of images that are more abstract than photographs, have more depth, and are more accurate than schematics. Drawing can provide spatial visualizations that could never be photographed or easily schematized. Together, we wanted to make some drawings of cargo being carried inside the cell. I started by studying dynein, a molecular motor that ferries cellular cargo from periphery of the cell towards the center of the cell. And I drew this form in rough sketches and in neat sketches, trying to look at it from multiple views to understand its form. But this motor really works as a dimer. So I started to look at the models which place them as dimers. And these a lot of these models based on circular figures and stick projections informed me very little about the differences in terms of the front and the back of the molecule, about the polarity of the molecules and the proteins that it associates itself with and how it carries the cargo. So I started to look at multiple data points, multiple models, and try to compile a summary in a very iterative process until I came up with this final model. And in this model, what you see is a summary of all the literature available until at that point of time. It shows not only what we know, but also what we don't know. And you see those details as dotted lines or fainter strokes. After understanding how the form perhaps is, I wanted to understand how the form looks as this molecule walks. Again, I went to the literature. Some data, including some from TFR labs, showed that the motor could either walk in discrete steps of 8 nanometer or 16 nanometer or 32 nanometer. They could walk in head-to-head relationship, which means that the one head always passes over the other, kind of like how we walk with our left and right foot. Some suggested that it could walk as it showed an inch-one walk, which means that one partner was always ahead of the other. I tried to plot these multiple permutations and combinations of their relationship with each other and the step size, and figured that there are various differences in how the relative distance between these two motors would be if they were to accomplish a walk like this. By then, some more information about how this dimer looks was available, giving me more clues as to how the dimer might rally. The general consensus in the field had been that the dimer shows a drunkard walk, which means it can take any number of steps. Any one motor can take any number of steps in the forward directions, in the sideway direction, in the backward direction. And while I made these drawings, I figured that while carrying a heavy cargo, as this does, it would slightly be difficult. So I made these multiple permutations and combinations of drawings in a more precise manner of the head and head walk, of the inch one walk with regular step size and with variable step size. And if I play them quickly, as I will do now, you will uncover that in the same number of steps, the amount of distance they can travel, and hence the amount of distance the cargo can travel, varies a lot. And what is also worth noticing is that the distance between these two motors varies quite a lot. For example, here the two motors are quite closely placed each time, whereas in something like this, the distance between the two motors is sufficiently large. And what it ends up meaning is that the tether to the cargo would have to stretch quite a bit to accommodate for the distance between the two motors. So through this drawing exercise, we came up with a lot of new questions. We asked, if both these components walk one after the other, how does the pouch move? Does it move after each motor has moved, or does it move only after both the motors have moved? We ask questions about the flexibility and the stretchability of specific joints within this protein complex. What is useful about drawing like this is that it is free from the pragmatic constraints of experimental feasibility. So now I'm gonna ask you to draw again. So this is an image picked from the internet. And I want you to think about, so this is the time zero figure. I want you to think about a time point t, and you can decide whatever is the change and whatever is the gap between time point 0 and your time point t. So imagine and explore, and you have 1 minute to come up with your hypothesis and share that with Kadambari via the email sayartspeaks@gmail.com.
Ipsa, I think you should perhaps explain what you mean by, develop this thing in time or something. Maybe it's not very clear what people.
Yeah, so I am right now here, and now I'm here. I moved. I'm here. I did this. I changed. So any change that should happen to this particular entity here, and the change could be anything you want it to be. It's a time for play and hypothesis generation. And the time could be 1 second, few hours, few days, few centuries.
This is almost like on Twitter, how it started and how it is now, right?
Yes, that's exactly what it is.
There is a request on the chat to repeat the activity quickly, if you could.
Sure.
And also, what is this? I don't know if you're going to tell them what is this, or people have to just take a guess of what it is.
Yeah, I don't want to. Yeah, so you can imagine whatever this is to I mean, it may not necessarily be the correct thing. This is the image, and you have to give it a change in time or space. And if this is something should happen to this, that's all you have to think about and make a quick sketch of it and share it at sciartspeaks at gmail.com.
So Ipsa, can you tell us if you are actually getting a few submissions or just passively people are listening? I hope people are submitting.
Yeah, there are several submissions. Towards the end, we'll try to scroll through all of them. I just couldn't figure that out properly while I was trying to do that in between the slides. Yes. But I have reports that, yes, people are submitting.
Okay.
All right, I'll give 30 more seconds. Those who feel shy of sharing, if you don't want to share, it's okay, but please do attempt the exercise.
I think whenever you show the work, you're not going to associate a name with it, right? So it's okay. I mean, we can be happily anonymous.
Yes, you can be happily anonymous.
That should encourage more people to send, I guess.
Yes. But if you want your name, you can definitely put in your insignia. That's cool too.
My drawing is still in the outbox.
Oh, no problem. All right. Let's move on. So any hypothesis worth pursuing may be realized verbally or visually. Yeah. I asked some science students and some law students to draw cells from memory. And here are some of the representative images from what they drew. Most of these images are flat cross-sectional views of a circular cell with nucleus right in the center. To challenge the status quo of how a cell is perceived, I devised a series of visual metaphors. In one, as I slice an orange, I slice a cell. And as I look into the piece of orange, I look into the piece of the cell. Audience, both untrained and trained, looked at this work and they could understand that a cell is not a 2D entity, but a 3D entity. They also recognized relationships between organelles, their distributions across the depth of the cell, the polarity of cell, even if these realizations were far from the jargons we normally use. We see, but we do not see. We use our eyes, but our gaze is glancing, frivolously considering its object. We see the signs, but not their meanings. We are blinded. We are not blinded, but we have blinders. With their scientific argument, Watson and Crick turned image of an X-ray diffraction result of DNA into a plausible stereochemical realization. And this model further applies to any DNA molecule. Each of its realizations stands for the rest. The molecule structure suggests a scheme of replication, a shift from description to explanation. Conceptually, it was easy to visualize how a single chain could be the template for synthesis of a chain with complementary sequences. While no one would claim that images are integral to all scientific thinking, it seems indisputable that scientific thinking, sometimes of the highest order, occurs in the absence of words. When probing into the unknown, drawing is an ideal tool because drawing is thinking, pointing. It makes things present and puts them at a distance. It is a mode of inquiry. Drawing can represent the seen and present the unseen, the known and the unknown. With this, I will conclude my talk and we will head on over to Twitter and see what you have drawn. Give me a second to set that up, please. All right. So these are some of the changes. This is of movement. This is of segmentation, movement. You can see, right? I'm assuming yes.
We can see.
Yeah. Okay, this is of damage. Okay, thanks, Sandhya. And these are the glowing worms. Cool. So glow is clearly represented beyond the body of the organism. Okay. This is warm. Milk. Colors. Okay. All right. All right. Okay. So I hope you enjoyed the drawing process and also looking at what other people think about worm, and about the changes that happen to that bug. All right.
So can you now tell us what was that object at t equal to 0 or whatever the time was? I mean, there was some bubbly thing with something in the middle of it on a green little, some rectangle on which there was some.
It was like bug waiting to emerge from its cocoon. It was a cocoon. Yeah. So yeah, with this, I thank you for your participation and your attention and your time and your invitation. These are the references in case somebody wants to read up, look up things about art and drawing and science together. And thanks to all of you. Thanks to Kadambri for curating the Twitter thread. Thanks to Minaj and my collaborators in Institute Cure with whom we did the dine-in work. And or to all the giants who have existed before us, who continue to pave way for us. And I am around. I am easily sort of accessible. You can find me easily on social media and on my website. Thank you.
Okay, and I hope you're happy to take some questions now.
Yes, absolutely.
Typically,
yeah, I should have said that. Yes, yeah. I'm happy to take any comments, feedback, questions. Yeah.
Yeah, so let's see on both on Zoom and YouTube. Okay. Ayush, do you just want to turn your mic on and ask the question?
Sure. Thanks for the lovely talk. And I just wanted to thank you also for like warming us into the drawing activity, because I feel like without that, the third activity would have been more difficult without the previous two.
Okay.
It was also pretty fascinating to just sort of like experience in the third one that it wasn't simply drawing, but to experience that inquiry process inside, right? Like we had to, like I had to think about like, is all the bubbly part of the organism, or is it like separate from it? All those things, right? In order to be able to do the drawing. So then I was thinking about like how in schooling, most of, at least my experience in drawing was that it's reproduction.
Yeah.
Right? Like there'll be a question, how is the cell, or like, draw a toad or a rohu fish or things like that. And we'll just have to draw by memory. And so I was thinking about like, but you are treating drawing as scientific inquiry, which is pretty fascinating. And so could you speak to the pedagogical use of this in classrooms?
Yes.
Or in out of class settings, but in learning environments.
Yes. So thanks for your comment, and I understand the context that you're trying to describe. I mean, we all came from the same education system, I suppose. But drawing as an activity itself, I think, has a great value. But how it is used and how it is encouraged, I think that has to shift a little bit. And if I'm not wrong, I understand there are people who sort of try to bring activity of looking and noticing and recording what you see as a part of the learning, activity and exercise. So I think the shift should be, what needs to change is the images in the textbook. I think they can be far better than what they are currently. And there should be space for looking and drawing and not just reproducing what's there in the textbook. So both of those changes can happen, should happen, and hopefully will happen, is how I think about it. Yeah.
Okay, are there other questions?
Can I ask a question?
Go ahead.
Hi.
Oza, yeah.
Hi, Ipsa. Thank you so much for the lovely presentation. I just had a question about accuracy when you talk about drawing in science, because I mean, I am an artist. I don't come from a very scientific background, but when I'm working with students who are, pursuing science, and let's say when I sort of do some workshop with them, for me, like, I'm quite open to this idea of the drawings not being so accurate, but as a scientist, how far is that limit of, accuracy when it comes to visualizing within science? Would like to know your thoughts.
Hello.
Hi, could you hear me?
Yeah. Yeah. So I could get part of your question, but I think I understand the question you're asking. So it depends on where that image is being used, what is the intent of that image, and what is the context of that image. So that's how I think about it. So if the image is to correctly represent, needs to correctly represent how the styrene molecule works, then the scientific accuracy needs to play a part. But if it is just an exercise in, it's not just like, but if it's an exercise in noticing, or it's an exercise in thinking, then we can lose some details. And it also depends on what you're trying to achieve. So for example, in the protein structure myoglobin image, what Irving constantly did was change things a little bit just to show what he needed to show to make us understand how this protein actually functions. So what he called selective lying. So even as a scientific illustrator, one constantly has to make tiny decisions, which sometimes either means loss in details or manipulation of the detail to finally give the picture that communicates the idea we want to bring forth. So I would say that the accuracy depends on what you're trying to achieve. If it's for proper documentation, then you might have to consider that more rigorously. But if it's about exploring, you can take in creative freedom as much as you need.
Yeah, thanks a lot.
Yeah, thanks, Ozzy.
There was a comment or question on the Zoom. It says, this is after your cell drawing. So for the cell drawing is deceiving as a 2D thing. Also for many other things, I guess. I mean, I don't know if this participant Jeannie or if he or she is there, maybe she can ask this question. But otherwise, if you understood what it is typed, you could also answer it probably.
Yeah, so this 2D thing is not a problem. Like we need to understand that what the 2D also looks like, because that's how we can look at all the structures that are inside the cell. But that shouldn't happen with a loss of realization that it's not like it's a 3D entity. So a lot of teachers would tell you that when students think about a cell, they think of it something like a sunny side up, like a flat thing, which has, the nucleus somewhere on top of it, which is the yolk part. So, so the perception that that image alone of the 2D cross section brings is incoherent and incomplete from reality. So that's where we need both images together, placed in a context where it's easy to understand that this is how it looks on the outside and this is how it looks on the inside.
There's a question from Alak. Alak, please go ahead and ask.
Yeah. So I'm not sure if I have properly formulated the question, but nevertheless, suppose you are looking at not just a drawing, but also a sequence of drawings or sequence of configurations, and you're trying to represent a process, perhaps through a movie. And we can take an example, for example. There is a very common process of synthesis of a protein molecule as the ribosome walks across the different nucleotides of a messenger RNA. And, in the same, and as the protein chain is actually folding, being synthesized, it also starts to fold, or at least to partly fold. And this was a much harder problem before the ribosome. I mean, before we knew about how the ribosome structures are and other things. But given the fact that you have a lot of data now, a lot of even understanding perhaps, is this process, how this thing happens, is this fully understood? Is there a representation for it? And if there is such a representation, where does one look it up?
That's a very specific question. I'm not sure, like that I have the best answer for it. But I could recommend a couple of names, especially if you are thinking about movies. Animators like Janet Iwasa and Drew Barry are some of the leading people who often compile 3D visualizations and animations based on them, relying on Janet Iwasa. And I can write that in the message if that helps. I just have to figure out how to go to everyone. Yeah. Janet Iwasa and Drew Barry. Drew Barry is in Australia. When you Google Drew Barry, you'll probably come up with some actress, but you're looking for the one who works at Vehi, Australia. So these people make a lot of 3D movies. The other person you can look for is McGill from DigiSign. He also compiled a book which is only available on iPad Pro. I haven't looked at the book because I don't have an iPad, but there's a possibility that something like that exists. Yeah.
Okay, there's another question from Amol. Amol, can you ask the question directly? Okay. Yes.
I think it's more of a comment than a question.
Hi.
Yeah, hi.
Okay.
Yeah, so my comment, as you see, is what's about this guy's drawing of protein. And it kind of reminded me of of paintings of cubist artists who would draw paintings from different angles. And I, of course, know, being very bad at drawing, never succeeded well in biology, but I wish that this was possible. A question that I was leading to was especially in chemistry, are there examples, for example, where attempts to draw drawings of structures has actually led to insights into actually knowing what the structure of a particular compound was?
So I'm not sure exactly in the same line with the thought process you have in mind, but I would say that the benzene story where he got the structure in his dream, that's also kind of like the visual happened before the discovery happened in some sense. Like so there, the visualization was an intricate part of how he realized the structure, what it was and what it could be. And the other example that comes to my mind is representations of different atomic models. I'm not really sure if the representations led to further thought by my direct study of that history, but I could imagine that the way the analogies were used to describe those models, like the plum model and so on, perhaps did influence how later models developed and thought about them visually. Like all of those languages have visual descriptions, which probably went on to be a part of the thinking process for later models.
Very nice. In fact, I also have something similar
Okay,
question.
I see there's, I mean, I have a follow-up question to that. I also was looking at something on the YouTube chat. Just connecting what Amol asked you recently with probably the first question, which inquired about pedagogy. And, I agree. The reason I dropped biology the first moment I could was because I was unable to draw the way they expected us to draw the cell or whatever it was in the book. And one of the problems I have is, for example, the working of the heart. I mean, I can make a drawing which enables me to understand how the heart works, even if it's just a perfect cube divided into 4 pieces, and then I put my appropriate valves in and out, which makes sense to me. Whereas the way they had the drawing in the book was completely confounding. So, are there efforts to actually simplify school level understanding of how biology works? Because today I think it's possible to really simplify these things in using the kind of sketches you've shown to make very simple drawings, which actually probably might be more or whatever, easier for people to understand, and you don't then expect people to be artists in drawing exactly how something looks.
Yeah, I understand it becomes a cognitive load for the student if the expectation is for it to be anatomically correct. But for the purpose of education, not for the reproduction in the textbook, I imagine that there is value in showing the anatomically correct heart and how its walls are placed within the anatomy. But as for knowing about an effort to make that simpler for the student, I am sorry, I am not aware. The direction that a lot of people are seemingly moving towards is not towards a generalization or simplification. I would rather say it's moving towards use of technology. So today, instead of having a cross-sectional view of the heart, you might find teaching supplemented with 3D movies, which go from outside of the heart to inside of the heart. So the efforts to help students understand are there, but I think it's in a different direction than what you articulated.
Okay, just there is a comment on YouTube. Dusty Hermit is asking that we love the orange slices compared to cell cross sections. Could you possibly show all the slices and how the comparison looks?
Oops. To find that in my, okay, let me just see. I can probably.
Yeah. Meanwhile, we can check if there are other questions. I don't know.
Yes. All right, let me see if I can share my screen. Can you see my screen?
Yeah, we can see your screen.
All right. So this is the slice 1, slice 2. So yeah, this is how the zine goes.
Okay, great. Thanks. Satya, do you see any other questions?
Not on this, and I actually it so happened that YouTube I didn't update, so I didn't see the questions. Now I can see. Yeah, but you have already. Asked. Actually, one of our colleague wanted to ask a question. Let me see if he's there. That's a Datar. Are you there? No, he's not here. Okay. Adab, I think there are no.
Yeah,
questions.
I think, and there are no more questions. So thank you so much, Ipsa. This has been a very, very different interaction. It's been fun to try to draw something. I don't think I've drawn anything like this.
Thank you.
So many
Yeah.
years. And thanks a lot. And we do hope you will come back here and we will have a more interactive session with hopefully a lot of our students.
Absolutely.
Yeah.
Yeah. Thanks for having me
Okay,
here.
over to you, Satya.
And yeah, if anyone has any feedback, please feel free to share over email or over social media, wherever you can find me. Yeah.
Sure. So on that note, once again, let me thank you on behalf of the Asset Colloquium to you, also your colleagues on the back end. I think you have helped it more interactive, and also, of course, to Arnab. Once again, thank you all. Thank you also all the participants. Very nice. In fact, looking at the comments on YouTube, it made very lively to see that many people enjoyed your talk, and particularly the interactive aspect of the talk. And with that, thank you. Thank you very much.
Thank you, everyone. Good evening.